There are estimated to be 5,000 to 6,000 viruses ready to jump from animals to humans, and an additional 1.5 million unknown viruses out there. The risk of infectious disease is growing as humans increasingly encroach upon the natural environment. These viruses threaten us with the potential of future pandemics, many of which may be more lethal than any we've seen before. We can prevent this.
This is a virus. Inside its shell is the genetic blueprint for making more viruses. It is 1/1,000,000th of an inch long, so small that 500 million of them could fit on the head of a pin. This tiny entity is devastating the world. This virus uses its spike protein to attach to a protein receptor on human cells like a key to a lock, letting the virus enter and infect the cell. Once inside, it hijacks the cell's processes to make 1,000 new viruses in 10 hours. These are released to infect other cells. 200 years ago, humans invented vaccines to combat viruses by killing them or preventing their entry into cells. As we speak, there is a new cutting-edge technology being developed, the mRNA vaccine. mRNA is a copy of DNA that takes genetic information to be made into proteins. When mRNA is injected as a vaccine, our cells use it to make the protein. Our immune systems learn to recognize the protein as an enemy part, and when the real virus enters our bodies, our defenses will be primed and ready. Moderna, among other biotech companies, is developing the first mRNA vaccine with mRNA that codes for the SARS-CoV-2 viral spike protein, and it has passed initial clinical trials with promising results. Conventional vaccines usually take 10 to 15 years to produce, but mRNA vaccines could be ready in a fraction of the time.
Science, engineering, and design have always drawn inspiration from biology and the natural world. Every organism on Earth is constantly evolving as they compete for resources in an evolutionary arms race. But what if we were able to look into the future and predict these evolutionary changes? Imagine the lives we could have saved if we had predicted what was coming. If we'd had policies already in place, vaccines ready to be distributed, and no need for a global lockdown at all. Just as we built optics inspired by butterfly wings and neural networks inspired by human brains, we can create software inspired by evolution. This is Viper, a biology-informed software tool designed to simulate the evolution of viruses. How is predicting the future possible with Viper? We use software very similar to it on a daily basis. Viper is like the predictive text on our phones. It uses deep learning, a kind of artificial intelligence that mimics the way a human brain learns. We give Viper learning materials to start with. This is the training data. For predictive text, this is all the sentences you write on your phone. For Viper, it's all the genetic sequences that contain the instructions for making the virus spike protein. With every new sentence or genetic sequence it encounters, the program gets better at recognizing emerging patterns in the data. Eventually, we can give it a small fragment of data, like the first couple words of a sentence, or the first pieces of a new spike protein, and it can fill in all the data surrounding it using the same patterns it saw during its training. In short, this is how Viper predicts new spike protein sequences. In addition, we are implementing a statistical model called a Markov model to examine the probability of nucleotides changing. It will complement the predictions from our deep learning simulation and increase the robustness of our tool. After we have our predicted spike proteins, we will test the key-to-lock interaction between the spike and our cell receptor proteins. The stronger the binding is, the easier it is for the virus to enter the cell, allowing us to choose the most likely and the most potentially harmful predicted strains.
So, what can we do with this information? Recall that mRNA vaccines are based off of the viral spike protein sequence. If we can predict this sequence before the virus ever emerges, then we can begin vaccine development in advance. The wait time for a vaccine can be devastating, and this is what the world is seeing right now. And this could be even worse in a pandemic caused by a more lethal virus. Instead of waiting for a high-caliber, pandemic-level virus to strike, we can begin vaccine research way in advance. mRNA vaccine development is estimated to cost between $100 and $200 million, which is really a drop in the ocean compared to the estimated $4.2 trillion lost so far in the United States alone. This is a wake-up call. We need to begin investing more in preventative biomedical technology. Viper is our team's solution. Even in the beginning stages of development, Viper is showing promising results. Based on early viral data, Viper predicted genetic sequences for SARS-CoV-2 spike proteins. These had over 99% similarity with spike sequences that appeared 4 days later in Luxembourg and 2 weeks later in Japan. Here, we compare a predicted spike sequence with the known spike protein from Luxembourg. Each axis represents the sequence position within a spike protein, the horizontal axis for predicted and the vertical axis for the Luxembourg one. The more similar the two sequences are, the longer the line fragments, and the better it matches a 45-degree line on the plot. As you can see, 3 long fragments of our prediction can be mapped exactly back to the known sequence, meaning our prediction is very similar to an actual spike protein. We then compare the different functional regions of our prediction with the known sequence. Functional sections specific to SARS-CoV-2 are present on our prediction. These results show that Viper was able to predict sequences that emerged after we trained our software. Imagine what we might be able to do as we improve it.
But could Viper's predictions lead to lost resources through development of unnecessary vaccines? Extensive consideration goes into developing each and every vaccine by virologists and policymakers alike to prevent the creation of vaccines that are not needed. And even if a predicted virus never arises, the cost of continued preemptive vaccine production would still be less than the cost of a single pandemic. And what if predictions of possible viruses are manipulated to spread undue fear? We can mitigate this by educating users on computational biology and the limitations behind our tool. Our team is already actively participating in efforts to inform the public about synthetic biology and its many applications. As part of our outreach efforts, we sought expert advice from a bioethics professor who suggested that biased and insufficient data collection could lead to inaccurate predictions. These errors will be minimized as more viral samples are collected, leading to higher accuracy in our predictions. What about the potential unequal access to preemptive vaccines? Socioeconomic disparity is a global problem yet to be solved. An economics professor specializing in poverty analysis suggested working closely with local vaccination programs to ensure that the produced vaccines can be distributed to rural and vulnerable populations. Since the production of mRNA vaccines is 10 times cheaper than regular vaccines, this could be achieved under special health acts at a reduced cost or for free.
A vaccine to end this pandemic won't solve the root of the problem. The risk of infectious outbreak is increasing as we destabilize Earth's equilibrium. We intrude on ecosystems by cramming wildlife into unsanitary conditions, decimating habitats, and forcing wild species to come into closer contact with us. Additionally, climate change pushes disease carriers to expand their territory. All of these increase the risk of transmission between animals and humans. H1N1, Ebola, SARS-CoV-2, and 70% of all human pathogens originate from animals. Scientists expect new viruses to emerge and for old ones to return with a vengeance. Although Viper was trained on genetic sequences from SARS-CoV-2, its framework can be expanded to predict the evolution of other viruses as well.
Envision a world in which we diligently monitor virus populations in wildlife, livestock, and people. We could predict when, where, and which viruses will cause an outbreak, and have a vaccine ready before it happens. What if this world was within our reach? As we grow aware of the myriad of ways in which our actions impact everyone on Earth, it is time for humanity to unite with a collective goal, to use our innovation to influence the world positively with our actions. A new world is emerging. A world that prioritizes biology.